Hello, everyone, and welcome to today's live broadcast, An Emerging Role of DNA Damage Repair in Huntington's Disease, presented by Animesh Ray, PhD, a professor at the Keck Graduate Institute. I'm Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen. Or you can use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Ray. I will now turn the presentation over to him.
Thank you very much, Judy, for the introduction, and it's a pleasure to be here. Let me start with a very broad theme. How do diseases occur? And you may already know that there are genes that sometimes make one susceptible to certain diseases. And so there is this idea of many variants, many genetic variants in the human population that have different degrees of susceptibility or resistance to any kind of disease. It could be metabolic disorder, it could be certain kinds of cancers, or it could be even infectious diseases. Some such genes have profound effects, and single genes are sufficient to cause a disease. And these are generally known as Mendelian variants or mutations in disease genes that segregate in the human population as Gregor Mendel told how genes should segregate. Then the question of environment. How does environment influence diseases? And they can be grouped into two classes: infectious agents, viruses, bacteria, protozoa, and many other organisms. And then there are the physical or chemical factors, pollutants of various kinds. There could be habit-forming chemicals, tobacco smoke, and things like that. However, it is not the case that such factors can be separated very cleanly into these two classes. What I want to emphasize is that diseases are the result of complex webs of interactions among genes, infectious organisms perhaps, and physical and chemical agents, pollutants, and other agents in the atmosphere. So the emerging idea is that all diseases are probably the net result of a large web of interconnectedness. If we look at the genetic variants that might provide certain kinds of susceptibility to diseases, there are two models, one of which is rather extreme, which is in the lower right-hand corner. And let us say that human beings have genes A, B, C, D, etc., a little over 20,000 of them. And there are many common variants of these disease genes that are segregating in the human population. And each gene has a few different variants that together comprise, the gene sets of the variants together comprise the particular susceptibility characteristic of any individual within the population. But there is a more nuanced model, which is on the top left, multi-allele hypothesis of diseases or disease susceptibility, is that of these, roughly 20,000 of such genes, there are many alleles, and these alleles segregate in the human population, and many of these are actually heterozygous in any individual. And the protein products, or sometimes the RNA products of these various alleles, would interact in complex ways to develop a susceptibility spectrum that is more than the sum of each of the parts. So diseases are complex outcomes of rather complex genetic signatures that all of us bear within our genomes. We came to this idea, and the rest of the talk will focus on Huntington's disease from doing something entirely different. My laboratory has been interested in understanding the properties of gene-gene interaction, and specifically how genome-wide interaction networks of genes are involved in maintaining the genome's integrity. That has been the theme of my work over the past 20 years or so. However, one of my postdocs in the lab at that time, who has now moved on, Biranchi Patra, while in the lab, was looking at the data obtained from a model organism that has nothing to do with human brain directly, and that is Baker's yeast. He noticed that a large number of genes that interacted with DNA repair genes were previously flagged because some of their homologs were interested in neurological disorders. Now, the kind of neurological disorders that were hinted at by these data had something in common, and that is they were all progressive diseases. All appear to be defective, at least in certain aspects of protein folding properties or protein folding machinery. And the examples are Alzheimer's, Parkinson's, a host of neurodegenerative disorders that affect the cerebellum and certain other parts of the cerebral cortex as well, but mainly the cerebellum, called spinocerebellar ataxias, and there are at least 30 of them, probably more, although there are only a handful of them are better understood than others. And ultimately, the Huntington's disease, which is a rare disease that afflict approximately 18,000 to 20,000 people in the United States, also a progressive disease. And people suffering from this disease become afflicted at the prime of their youth generally, but the disease can also affect very early on. The earliest person who had developed clinical manifestations of the disease was only 4 years old. This disease is a inherited disorder, and as this particular pedigree shows, that it is inherited as a single autosomal dominant manner. And that is how this disease was first discovered, being inherited within the human population. The dominance is because of a mutation in the gene. The gene maps on the left end of the human chromosome 4. And if you look at the encoded protein, the protein is pretty complex. It has got a large number of very interesting features. For example, a polyglutamine or poly-Q repeat near the left end, which is the N terminus of the protein. But then there are also other repeated units. For example, a little bit to the right of poly-Q, there is a polyproline stretch, a stretch of amino acids that are enriched in proline. And then there is a repeat that is called a heat repeat, which is found in a class of proteins that have this particular repeated structure. The heat repeat motif is actually repeated at least 3 times that we can recognize. And near the C-terminal end, the carboxy-terminal end, there is also a polyglutamine, polyserine, and polyproline stretch mixture of these 3 amino acids. The problem is that we do not fully understand even the normal function of the protein. But we know that if the gene is mutated in the sense that the polyglutamine repeat in the N-terminus is enlarged from the normal range of polyglutamine, which is generally about, varies between 15 and 28 to 29 glutamines. If it is enlarged beyond 37 to 38, at least beyond 40, then one gets a disease. And the gene that contains this repeat is therefore a gene that has a repeated trinucleotide sequence, CAG. CAG codes for glutamine. So there is a long repeat of CAG, CAG, CAG like that in the normal gene, approximately 25, on average, 25 to 28 CAG repeats. But then in the people that suffer from the Huntington's disease, this repeat extends to 40 or more generally. Now, if you look at the Huntington's disease gene, and here abbreviated as HTT, and the protein, by the way, has a colorful name of Huntington, the gene is divided into 67 exons, and the polyglutamine repeat, the polyQ repeat, is only at in the first exon. So if you look at the protein, which has a complex structure, it appears that the disease is mainly related, not exclusively, but mainly related to the presence of a long stretch of polyglutamine only encoded within the first exon. However, a recent work reported last year shows that there might be an abnormal processing of the pre-messenger RNA out of this gene, which terminates either within the second exon or within the intron, or within the within another place within the first intron. There are two sites that can serve as abnormal transcription termination sites, and there is a claim that this particular abnormal transcription termination may be related to the production of an N-terminal fragment of the normal full-length protein that contains the CAG repeats expression in terms of polyglutamine. And it is this abnormal transcript, that much shorter transcript, that when translated into this short fragment of protein that contains the long stretch of polyglutamine, is the toxic agent that causes this disease. The gene, as shown in this slide, is expressed in nearly all cells. In the brain, which is shown in the yellow boxes, in the brain it is expressed very highly, especially in the cortical cells, but it is expressed in nearly every tissue that has been examined. The cell line that shows the pink box way up, that actually is an artificial expression, so we should discount that. But other than that, these box plots represent the level of the messenger RNA in various tissues that are labeled below. So you can see that the log ratio of the fold ratio of the mRNA is actually high in nearly every tissue that one examines. Despite this fact that either the normal protein or the abnormal protein is expressed in every cell, the disease hits only certain small group of cells within the cerebral cortex. And that is shown in the next slide. The medium spiny neuron are the first ones to degenerate in the brain of the Huntington disease patient. And later on, higher cortical cells, such as the cortical pyramidal neurons, also begin to degenerate. And this slide is complex. It summarizes what we understand about what happens when the abnormal polyQ containing protein is expressed in the medium spiny neuron. For example, the protein is known to misfold and also catalyze misfolding by abnormally interacting and perhaps saturating the proteasome complex, which leads to mitochondrial dysfunction that is supposed to trigger cell death. The protein is also known to go into the nucleus and interfere with the gene expression. The full-length protein is not supposed to go into the nucleus, but generally only a fragment of it is found to accumulate within the nucleus. And it is the abnormal protein, that is the protein fragment, that contains the long length of the polyQ repeat seems to be abnormally interfering with the gene expression of many genes, including certain genes that are important for neural function, such as derived neurotrophic factor, and so on. The protein also appears to function within the synapse, and it has been implicated in transport and translocation of proteins and RNA cargo along the neural filaments onto the synaptic terminals. And it is thought that the abnormal protein interferes in this synaptic transmission as well. So the protein might be doing a whole lot of different things. At least the toxic protein might be interfering with a large number of different molecular processes: mitochondrial function, proteasome function, gene expression, axonal transport of materials, and reuptake of neurotransmitters at the axonal junctions. Now, a lot of researchers who are working on Huntington's disease look at it from different perspectives. And we are like blind men who are trying to interpret what we are looking at while touching various parts of an elephant, whereas the real elephant lies hidden behind high levels of complexity. So to summarize, it appears that the CAG repeat length within the first exon of Huntington's disease gene, or Huntington gene, is very important. Within the germline of certain individuals, the CAG repeat length appears to expand so that their children are born with long tracts of CAG within the first exon, which leads to expanded polyglutamine tract within the Huntington protein. And people with repeats 36 or less are generally normal. People with CAG repeats of tract length above 40 are 100% afflicted by the disease. However, how early the clinical manifestation of the disease will set in depends on how long the tract is. If the tract is short, like 37 to 40, the disease afflicts much later in life. On the other hand, if the tract length is very long, about 40, the longer it is, the earlier one gets the disease on average. It is thought that the abnormal Huntington's disease protein is toxic to the median spinal neuron, but it is also toxic to a large number of different cells that have been looked at, especially in model systems. For example, yeast, the baker's yeast, expressing the wild-type Huntington's protein fragment, N-terminal fragment, is perfectly normal. But the same N-terminal fragment containing a polyglutamine repeat, in a long polyglutamine repeat, like 40 repeats or longer, the yeast cells die. Similarly, if the human normal protein of different kinds of versions are expressed within the Drosophila eye cells, which have part neuronal characteristics, the eye cells degenerate. If the protein is the mutant protein, but the normal Huntington protein does not affect the eye cells. Similarly, in the worm C. elegans, which is another beautiful model system for neurobiology, if the protein is expressed in the body wall muscle cells or in certain neurons. If the protein is normal length polyQ, nothing happens. But if the polyQ stretch is long, then the muscle cells or the neuronal cells show defect, and the worm shows all kinds of phenotypes, such as faster aging, sluggishness to touch response, and the ability to move around is made sluggish. In other words, the worm also shows various kinds of behavioral and mechanical phenotypes, abnormal phenotypes. If you look at human beings that contain, the patients that contain long expanded polyQ repeats protein expressed in their brain cells, you see that progressively the person would develop neurological symptoms. It is thought, speculated, that most likely the abnormalities, in other words, the toxic effects of the proteins, set in pretty early, but they're simply undetectable by the present day diagnostic techniques. But when the clinical manifestation occurs, it's probably already too late, because the toxic levels of the protein has already done damage to the brain cells. And various degrees of clinical manifestation, such as very early signs such as depression, clinical depression, and then later motor impairment, cognitive impairment, and dementia, followed ultimately by paralysis and chorea, which is involuntary movement of the extremities. These things happen, and this is also correlated with changes in the macroscopic changes in the brain. For example, the cortical gray matter size is depleted over time. Cortical white matter is depleted faster, and the striatal volume also collapses. So there are two models as to how the molecular pathogenesis occurs. One is a simple protein accumulation model, and that assumes that the rate of progression of this neurodegenerative disease is entirely dependent on the mutant Huntington accumulation over time. So the longer one waits, the more such mutant protein accumulates. If you think that the polyQ length is constant within a particular patient, let us say a patient that contains a gene with only 50 CAG repeats is going to develop the disease somewhat late in life, let's say around the middle age, around 45 to 55 years of age. And that is because the protein containing approximately 50 Q repeats, glutamine repeats, is going to have a tendency to accumulate, and that accumulation tendency is such that critical clinical symptoms arise around the Middle Ages or so. However, a person bearing, let's say 80 CAG repeats, in other words, a polyQ of length 80, is going to have protein accumulation, the toxic protein accumulation in the brain cells much faster. And so this person is going to have, most likely on average, have a much earlier onset of the disease, let's say by 24, 25 years of age or so. The second model says that given that the person is born with at least one copy of a particular long repeat length at or above 40 CAG, there is a tendency in the somatic cells of the body, including in the brain cells, during the replication of the pre-neuronal precursors, for the somatic expansion of this CAG repeat within the cells. Sometimes the repeat length may contract even, but sometimes it can expand. This leads from the observation that people with approximately 34, 35, 36, 37 repeats tend to have children who very often have much longer repeats. Sometimes the children have shorter repeats as well, but they do have children that have longer repeats. So if it can happen in the germline, can it happen in the somatic cells? So a person born with either a mother or a father, usually a father, that contains, let's say, 40 repeats, in the germline of the father, the 40 repeat of CAG may expand to 60, and then a child may be born with 60 repeats. Now, it is possible that this person, during the cell division in embryogenesis and brain development, and then later, some stem cells in the brain, can actually have unstable repeats. So the repeat length may increase even faster, may increase even higher to, let's say, 70, 80, even 100 or more than 100 CAG repeats. And in these cells, the toxic protein is very highly toxic because it has got so many Q repeats. So this model says that these somatic cell heterogeneity might predispose the person to much earlier onset of the disease than otherwise possible, having only, let's say, 40 or 50 repeats to begin with. So in other words, polyQ length of this toxic protein varies among the neurons, and the neurons with longer repeat lengths will degenerate faster. That's the assumption. However, let me make it clear that the two models are not mutually exclusive, and both of these two processes might be happening to some extent. A recent publication sort of summarizes these two models together and explains that the adult onset repeat range, which has shorter, relatively short CAG repeat length, is explained by the fact that shorter polyQ proteins might be less toxic than longer polyQ repeats, whereas those people that have a much earlier onset of the disease, such as the juvenile onset repeat range, they have much longer repeats, and that could be because the protein containing longer CAG, protein containing longer polyQ repeat may be much more highly toxic, or perhaps the repeat instability, the somatic repeat expansion, explains the early onset in such people. I have mentioned that the repeat length is inversely proportional to the age of onset, but that is an oversimplification. If you look at the real data, the data are sort of messy. Yes, you can find a line of regression through them, but there is a large scatter in the data. The variance, about 70% of the variance in the age of onset can be explained by the length of the CAG repeat. However, and this length of the CAG repeat is only guessed at not by the length of the CAG repeat in the brain, but length of the CAG repeat in the somatic blood cells. So it is not clear whether the length of the CAG repeat in the blood cell exactly reflects the length of the somatic of the CAG repeats in the brain cells, because there could be somatic recombination in the brain. Nonetheless, it appears that from this data that although on average it is true that as the repeat length increases, the age of onset is earlier, there are a lot of exceptions. For example, somebody who may have a clinical symptom very highly developed at the age of 50 may have a repeat length varying from 40 to about 55 or even 60. So there is a lot of variance, and about 33% of the variance cannot be explained by the repeat length only. And so one begins to think of other influences, such as environmental or even other genetic factors that are inherited in human population because of the variants in the 20,000 other genes, 20,000 or so other genes that are segregating in the human population, the different alleles that I alluded to in the first few slides. So this is diagrammatically explained in this slide. So CAG of length 40 has a much later onset. CAG of length 80 has an earlier onset, and CAG of 100 has a much earlier onset. However, if you choose people with, let's say, an average CAG repeat length who manifest Huntington's disease, say CAG of 50, then you can imagine that there might be other alleles of some of the 20,000 other genes which might be good modifier genes. And therefore, they could push the age of onset of clinical manifestation to a later date. Or there could be some other alleles that are bad modifier genes that could push it to an earlier onset date. So if so, we ought to be able to discover good and bad modifier genes for Huntington's disease. And what could these modifier genes be? Well, the modifier genes could be any of the genes whose products interact with the Huntington's disease protein, the normal or the mutant. The normal Huntington's disease protein is actually essential for embryonic development, especially for brain development. So it is an important part of brain development. It is not clear whether the protein plays any role in other cell other than the brain. And in fact, evidence suggests we don't know for sure, but at least it is important for mouse and human brain development. And either interacting proteins with the normal Huntington's disease protein or with abnormal Huntington's disease protein, if there are allelic differences in the genes encoding such genes, they might be good or bad modifier of the disease. So let us try to look at it in slightly more detail. So let us say that a mutant protein somehow produces a toxin. And there is a modifier gene X, which needed for the mutant protein to make the toxic effects possible. So if such a modifier gene mutated so that the gene is lost, let's say deleted, then such mutations would suppress the Huntington's disease manifestation because the toxic effects will be milder because it is not there to help the mutant Huntington protein to produce the toxic effect. In other words, such mutations would show up as suppressors of Huntington's disease phenotype in model systems, for example. And in humans, they might, certain allelic variations that would reduce the activity of the protein can show up as good modifiers of Huntington's disease. If we can identify such genes immediately, such gene products could be plausibly therapeutic candidates because by raising a small molecule, we might be able to inhibit the activity of the gene X's protein and thus would basically phenocopy or rather translate the effect of the mutation in such gene. So we would make the protein crippled by means of a chemical, and thus the mutant Huntington protein will not be able to exert its toxic effect. There could be the opposite kind, and that is there could be a modifier gene Y which normally suppresses or inhibits the mutant Huntington protein's toxicity. And therefore, loss of function mutations of such modifier gene Y would take away such negative influence and therefore would enhance the toxic effect. So the loss of function mutations in gene Y would enhance Huntington's disease. And you could imagine a gene Z, which is a negative regulator of the modifier gene Y, will look like the modifier gene X, and therefore that also can become a therapeutic candidate. So now you begin to see a picture emerges gene-gene network being important in understanding and perhaps discovering therapeutic candidate targets for Huntington's disease treatment. As I mentioned before, we came to this problem of Huntington's disease by studying a completely different question. We were trying to understand how gene-gene interaction network maintains the integrity of the genome. And in order to do that, we mutate some essential gene in the organism, so the organism dies, and then we manipulate other genes in the genome so that the organism is resurrected, so to say. And one of the ways we could resurrect the organism by bypassing the requirement of some essential gene is by means of rewiring the gene interaction network. So imagine a pathway on the left that produces some gene product X through a series of gene actions, E, F, and so on. And this X pathway is essential for life. Now, suppose a mutation occurs in F so that the pathway X is inoperative, and as a result, the organism dies. What can we do to resurrect the organism? What we found is that there exists a large number of either structurally or functionally redundant genes in the genome that we can tweak in order to resurrect the pathway X. Some of these genes, such as G or H, normally do not participate in the production of X, but produces Y, which may or may not be essential for life. However, in the absence of the gene product F, we can perhaps tweak the expression of gene G, which is a structurally redundant gene for gene F, makes a protein that is very similar but not identical to F, so-called paralogous genes, may take over the function of F and thus rescue the mutant F. So X, the pathway X, becomes operative again, and the organism survives. We also found a much larger number of genes of the type H, which have no structural relationship to any of the genes that way of effect, but they're functionally redundant. And they can be, their expression can be tweaked in series so that they take over the function of the lost function of F and thus resurrect the organisms through restoring the pathway X. Such genes are functionally, not structurally, but functionally redundant, and they are called degenerate or promiscuous. They catalyze degenerate or promiscuous pathways. So structural and functional redundancy appear to be important for maintaining genomic integrity. However, there is a paradox in here. Imagine that one needs to cross a stream from one side to the other, and there are 3 bricks or stones on which one can step on. And these 3 bricks are like 3 genes. They're essential. Now, suppose that you put a non-essential gene, like a plank, on top of this. And now you can walk over the plank. But the plank is not essential because you can hop over the, you can basically step over the bricks. But if the plank is there, it is possible for the evolution to actually mutate away one of the 3 bricks, the one in the center, because it has no function. So you can walk over the plank from one side to the other. However, now one of those bricks have become essential because the span between the extreme 2 bricks is very long. And if that brick is taken away also, the plank collapses. And as a result, you have a non-functional bridge. And similarly, you have a non-functional pathway if an essential gene is removed. In other words, if you have genes that are essential, it introduces vulnerability to the organism in terms of whether the genes can be mutated and still keep the organism safe in terms of disease susceptibility, so to speak. So this is the concept that we have been very interested in for a very long time. And we found such functionally redundant pathways that they are cryptic pathways. They exist in the genome, a large number of them, and they are responsible for functionally reconfiguring gene-gene interaction networks to take care of mutations in essential genes. One such example is shown here in slightly more detail. When a cell divides, there is a trigger that induces cell division. Cell cycle begins and goes through the process of mitosis, ultimately, and then mitosis begins. At that point, DNA condenses to chromosomes, distinct chromosomes, and then the chromosomes separate to the two daughter cells. This process is dependent on a particular protein called SMC2, and if you mutate away the gene for SMC2, it blocks that pathway. The DNA fails to condense, and as a result, the chromosomes fail to separate. The DNA is ripped apart during the cell division process, and both of the two cells die. However, it looks like it is possible to bypass this in two entirely different manners. In one manner, you can prevent the DNA to get entangled even though the DNA fails to condense. And by the second way, you do not prevent the DNA entanglement. The DNA fails to condense, they entangle. However, you can facilitate the DNA disentanglement by other genes, and thus bypass the mutational block of SMC2 and then allow the chromosomes to separate and the two daughter cells survive. This is the kind of story we were interested in. And Biranchi in my lab found that a large proportion of genes that could bypass the SMC2 block appears to have been flagged as those somehow involved in neurological disorders. So we went to yeast genetics to actually probe this question. Are there genes that could bypass the Huntington's disease gene toxicity? Huntington's disease being a model of neurodegenerative disorders. And this was started way back about 20 years ago by Susan Lindquist, who unfortunately is no longer with us. She had shown that you can use the awesome power of yeast genetics to actually probe this. So you can look at the physical interacting proteins of the wild type and mutant Huntington's protein. You can use yeast genetics to probe such interactions as genetic suppressors or enhancers of the wild type or mutant Huntington protein activity in yeast. And the goal is to generate hypotheses for future testing and validation in other organisms. So Biranchi did that. He expressed the normal Huntington protein in yeast and the cells survive. He expresses the mutant Huntington protein in yeast and the cells die. And now he expresses candidate suppressor genes in the cells that express the mutant Huntington protein and asks whether the cell dies or lives. And by this means, he found a large number of suppressors and enhancers of the mutant Huntington protein, which people did not discover before. And some of these findings led us to suspect that certain neurotransmitter metabolism, for example, kynurenine, which appears to be a very important metabolite in the production of a large number of small molecule neurotransmitters in the central nervous system, its metabolism is somehow related to the integrity of chromosomal DNA, genomic DNA. In this slide is shown a network of the genes that link the SMC2, SMC4 condensin complex to the kynurenine metabolism genes BNF5 and BNF4, and how the human Huntington, the abnormal protein, somehow interacts with the chromatin reorganization complex SSC2 and SSC4 to stabilize the condensing complex and interact with the human with the yeast genome that somehow protects the mutant Huntington's effect through the kynurenine metabolism. The proposed connection between the kynurenine monooxygenase, an enzyme required for metabolism of kynurenine, and mutant Huntington protein is diagrammatized here, in which we suspect that BNF4 and BNF5 proteins together make a complex, which somehow interact with the Huntington protein, and the mutant protein interacts in a manner such that the condensing stabilizing complex during the mitosis of cells or during other processes within the cell. For example, neuronal cells do not divide, but pre-neuronal cells do, and the neuronal cells require condensation of certain parts of the chromatin in order to express certain genes, such as the ribosomal DNA and various other DNA. And these processes may be abnormal through this particular interaction scheme that we have proposed here. This slide shows in more detail some of the mechanisms in which other DNA repair recombination proteins, such as RAD51, MND1, and DMC1, and we have genetic evidence for these, somehow interact with the kynurenine metabolism involving mitochondria, and how the Huntington protein is interacting with the various players of the chromosome dynamics and DNA repair recombination to screw up the process of kynurenine metabolism and mitochondrial function. So we wrote a paper suggesting that genetic modifiers of Huntington's disease that also function in maintaining the genome integrity might be important to understand the pathophysiology of Huntington's disease. And so we went to the computer to try to investigate this process. So the idea is that in America, rather than and not in England, you are who your neighbors are. In England, you are who you're born from. So in America, because you are who your neighbors are, you look at the proteins that interact with each other, and by looking at the identity of the proteins in such protein-protein interaction network and knowing some of their functions, you can the complexes may betray the function of the unknown proteins. So this is what my PhD student, Sonali, tried to do. She is a medically qualified person. She has an MD, but she came to my lab to do computational biology. So what she did is took the gene-gene interaction data that we generated, took genome-wide loss of function suppressor screens in yeast for mutant Huntington protein toxicity of various sets, some done in 2005, some done in 2003. And then we did a separate work in 2003 in which we found other suppressors of the Huntington's toxicity. And then she combined these data of gene-gene interaction with data sets of protein-protein interactome obtained by mass spectrometry of co-immunoprecipitated proteins from mouse brain. So she took the genetic interaction data from yeast, took the protein-protein physical interaction data from mouse brain, and then put them together in some clever ways, and then mapped them to the human protein-protein interactome, which is deposited in the HIPPY database, and found a large network of interactions, which she called the Huntington's disease integrome. 16% of the total protein-protein interaction in the human interactome mapped onto this Huntington's protein interactome. So are the Huntington protein interaction partners in the human inferred interaction diagram network, do they imply related function? So how do you ask such a question? So what you do is a series of machine learning. You apply a series of machine learning algorithms. And one of them, this is a class of unsupervised machine learning algorithm, so-called clustering, which captures overlapping functional relationships among genes and proteins in the network. So imagine two genes, 1 and 2. Are they related? Well, they would be related if the product of gene 1 interacts with, let's say, some other proteins, F1, F2, and F3, and F4, and the product of gene 2 interacts with F5, F6, F7, and F3. So if two genes share one interactor, they're related by function. So this is what I meant by you are who your neighbors are. So using an algorithm called cluster 1, which looks at protein-protein interaction network based on some cohesiveness measure, which I will not go into this, the technical details of it. She ran it under the logic that the yeast gene-gene interaction somehow has an evolutionary relationship to the mouse protein-protein interaction, which has an evolutionary relationship with the human protein-protein interaction. And such relationships have precise physical meaning. For example, if gene 1 interacts with gene 2, then it may be that product of gene 1 may actually regulate the expression of gene 2, or the product of gene 1 and gene 2 physically interact with each other to make something meaningful, or maybe a combination of these two models. So she applied such clustering algorithms onto the integrated interactome of Huntington's disease integrome, which looks like a big mess of spaghetti. However, machine learning, unsupervised machine learning, which allows people to do clustering of clusters of network members on the basis of some hidden properties that are not very apparent in the beginning, allows one to discover meaningful associations. But there is also another method called supervised learning, which is able to classify the mechanisms of such hidden rules within the spaghetti of interactomes, and I will discuss that. Let us concentrate on the unsupervised learning first, in which the protein-protein interactome is classified based on connectivity and density functions, which led to several very statistically highly significant clusters. And some of the top clusters had proteins that were enriched for functions of DNA damage, DNA repair, and chromosome organization. This was surprising because we did not start with any hypotheses about what the cluster, the neighboring functions would actually imply. We simply took the entire data sets and ran the clustering, found the statistically most significant clusters, and lo and behold, what came out? Our DNA repair function. Again, another DNA repair function. So this is not a fluke of the clustering method, because a second clustering method also gave rise to essentially similar results. And I will not go into the detail of this. So having run these clustering algorithms and having found these protein clusters that may have something to do with DNA repair recombination in interactions with the mutant Huntington's protein, we wanted to see how far this method can be generalizable by trying to validate the candidates of the clusters in other model organisms. So we went to Drosophila and took the first 20 to 25 genes, of which we have so far been able to test only about 10. And out of this, 1, 2, 3, 4, 5, 6, 7 candidate genes out of 10 have turned out to be actual modifiers of mutant Huntington protein on the Drosophila I phenotype. For example, NPLock4, ESPL1, NAPC7, and so on, in LRC4, all of these genes, either the loss of function of these genes or the gain of function of these genes either enhance or suppressed the effects of the mutant Huntington protein. Stop for a second and think about what we did. We took genetic interaction data which suppressed mutant Huntington toxicity in yeast, combined them with a host of mouse brain data in which the mutant Huntington protein was co-immunoprecipitated, and by mass spectrometry asked what other proteins came with it, combined them using evolutionary criteria, and mapped them on the human protein-protein interactome, clustered them to find what functions there are. The top functions had something to do with DNA repair recombination. We took those human genes, found their Drosophila orthologs, and then mutated or overexpressed some of these genes. And lo and behold, what we find is that at a very high frequency, 7 out of 10, we find genes that express some modifier function for the mutant Huntington's toxicity. Very interestingly, as we were doing this experiment, we came across a paper published in Cell back in 2015. And when we did this work, the paper was not published yet. It did a genome-wide association meta-analysis of a few thousand people in Europe and America combined, tried to see whether there are variations among human population in other genes that might provide a clue to modifier loci within, for Huntington's disease patients, age-of-onset phenotype, and reported that there is one gene that they found to have a high signal. This gene was called FANCI, F-A-N-C-I, Fanconi's anemia complex group 1. This gene was previously known to be a Mendelian disease gene for Fanconi's anemia, which is a disease of the blood circular system of the connective tissue, a hematological disease. But its gene seems to, some alleles of this gene appear to be a modifier of the Huntington's disease age-of-onset phenotype. The gene was known already to have something to do with handling of the DNA repair process, and that seems to have something to do with the modification, both the Fanconi's anemia and modification of the propensity for getting early onset Huntington's disease in humans. It was also shown to be a modifier of spinocerebellar ataxia of a certain kind. So it looks like we have hit a jackpot that already in our candidate, which we haven't tested yet in any other organism, there's a human gene that has been reported from a large genome-wide association meta-analysis to be a modifier of Huntington's disease. So let's go to the second method, supervised learning. So in supervised learning, one takes a raw interaction data and then manipulates that to make a scaled data and divides them into two sets, a so-called training set and a validation set. And the training set is used to instruct a model that we supply. And the computer, the machine, learns from this training set some hidden association within the data, and then spits out a model, a trained model, which we validate using the validation set, and then using a completely new data, ask what is how efficiently the model is validated. So we get a profit. You can easily imagine where we got this picture from the internet, from finance companies. But actually, we are not in the business of making money. For us, the profit is the genes, the identity of the genes that provide the hidden relationship with the mutant Huntington's proteins modifier. So Sonali ran a host of such supervised machine learning algorithms and explored their ability to classify the data and to simplify and make the long story short. She found one that actually predicted with very high efficiency, over 85% efficiency, new interactions that the machine did not know before. And I'm going to go through this very quickly. So we had to supply the model. So what model? So we said that certain features of the protein-protein interaction network in the Huntington's disease interactome, abstruse geometrical models, mathematical models of the network. These are features in the learning parameters, as well as protein domains and motifs. As it turns out, so what do explain in the next paragraph, in the next slide, that the data are of three types. The training sets are of three types: proteins that interact with the wild-type Huntington protein, proteins that interact with the mutant Huntington protein only, and proteins that interact with both wild-type and the mutant Huntington's protein. And using these three data sets, can we instruct the machine to learn something about it so that they can classify new proteins in these three groups. And geometrical properties and protein structural properties that we used: protein motifs, protein domains. There are 96 motifs and 673 domains. And then a series of abstract geometrical properties being protein interaction network. And I will not go into the definitions of these, but suffice it to say that what she found is that protein domains were not at all instructive. It is somehow the protein motifs, but mainly the abstract properties, geometrical properties of the protein-protein interaction, such as betweenness centrality, closeness centrality, and things like that. They were the most important parameters to train the machine to be successful. This is how the input data look like. For the brevity of time, I will not go into this. Among the various models, machine learning models that she used, only the GBM or gradient boosting model was more efficient than others. And ultimately, the GBM results show, the one in the center, is that area under the curve of 0.85 on unknown data, which basically is a pretty high accuracy of predicting novel interactors of mutant Huntington protein. The interesting thing about supervised machine classification is that it can also regress, which means it can think backwards or compute backwards to find the modifier genes themselves. So what did we learn? We learned a host of modifier genes, which are listed here, too small, I suppose, in this slide, but I will not go into this detail one by one. But most of these genes have something to do with already known human neurological disorders. Stop for a second and think about what we did now. We imagined that there are some hidden properties of proteins that interact with a mutant Huntington protein and trained computer algorithms to search for such hidden properties. And once the machine was trained, it could identify such features and told us a set of proteins or genes that should with high probability interact only with the mutant Huntington protein. And lo and behold, some of these genes are already known to be involved in certain progressive human neurological disorders, not Huntington's disease, but other neurological disorders such as spinocerebellar ataxia and certain other diseases. So we are now taking this model system to another organism, taking these inferences to another model organism, and that is the Caenorhabditis elegans, which has exactly 302 neurons compared to Drosophila, for example, which has some 250,000 of them. So it's a much more tractable organism. And as the diagram below shows, that majority of the neural connections are already configured for this organism. So we can begin to ask very precise questions about whether some of these genes actually modify the Huntington's disease protein's toxicity in the worm nervous system. And we are collaborating with Paul Sternberg at Caltech to do this work. Now, last January, Craig Venter and his co-workers published a paper in Nature Review of Genetics where they scanned tens of thousands of genomes and in vitro experiments for genes that are essential for human survival, and came up with a core set of 188 essential human genes. And they showed a gene-gene interaction map of these 188 essential human genes. Amazingly, many of our candidate modifier genes of Huntington's disease protein map onto this particular 188 essential human gene interaction map. They have shown, Craig Venter and his co-workers have shown that there is a very high likelihood that mutations in one or more of these 188 genes occur in people with diseases, genetic disorders, as this diagram taken from their paper suggests. So the pathogenic variants of essential human genes at a very high frequency predispose the human population to certain genetic disorders. And we have shown that the computationally identified set of modifier genes are highly enriched in terms of their membership within the 188 core essential human genes. So the question at last is, does the robustness property of the genome make us intrinsically vulnerable to diseases? The genomes are there, presumably for the selfish reason for the genome to be propagated. And therefore, there are reconfigurable networks that have been selected in evolution. But the other part of this is that such reconfigurable networks may have made the hosts of such genomes, in other words, us, vulnerable to diseases. The genomes are selfish, they don't care. But as a result, we become vulnerable to genetic disorders. However, can we exploit this very vulnerability to find cures for diseases, such as the modifier X that I had mentioned a few slides back, which could be a drug target? And that is the intent of our future work. So from a fundamental question, which is the basis of responsive gene interaction network, we did some experiments using serendipitous models, found by chance a disease connection with the idea that networks embody information. We learned the hidden knowledge in large data sets, and we let computing machines learn from the data. With model organisms, we are evaluating whether the machines are learning well, and would the fruits of the machine learning make biological hypotheses about the disease testable in the future, and perhaps give us leads for discovering novel therapies. So in the closing, let me acknowledge the work of Biranchi Patra, who has since moved on, Jesse, a previous graduate student, a PhD student who had also moved on, Sonali, also moved on, Katerina Venderova, my colleague, and her laboratory, and Radek in Keck Graduate Institute's School of Pharmacy, who have done all the fly work. Dr. Songjun Koo, who is a collaborator, he's an applied mathematician. Paul Sternberg in Caltech, with whom we are now collaborating using the worm system. And innumerable KGI students, including undergraduate students from outside who have worked in my lab over the past 14 years. Thank you very much. So we can now have further questions if you have any.
Thank you, Dr. Ray, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click on the Send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, which of the two models, cumulative aggregation of one polyglutamine length protein or somatic instability of the repeats, explains the computational findings?
That's a pretty good question. Unfortunately, we don't know the answer to that, but let me try to explain some implications of that. So DNA repair recombination processes not only repair damages to the DNA, they're also known to actually participate in the processes that expand and contract repeated DNA. There are decades of work on that. So the fact that modifiers of mutant Huntington protein are sometimes participants in DNA repair and recombination can work in either way. In one way, somatic recombination might be enhanced such that long repeats can be produced from shorter repeats, and as a result, bad modifier effect be actuated. Or the recombination enhancement can can contract the repeat length, and so good repair function can be actuated. On the other hand, entirely differently, it's possible that the toxic effects of the mutant Huntington protein somehow manages to damage the DNA. This is hinted at by Biranchi's work, which shows that DNA chromosome proteins physically interact with the mutant Huntington protein. So the mutant Huntington protein might interfere with the chromatics DNA decondensation and condensation process, may lead to entanglement of the chromosome of the DNA, and thus interfere with gene expression in neural cells. And as a result, the genomes within the neural cells might collapse, might disintegrate. And perhaps some of this is also happening. So the computational work is agnostic in terms of what mechanism by which these modifiers are acting. So now the time has come to painfully address these questions one by one to find the actual molecular mechanisms by which the mutant Huntington protein interacts with the DNA damage repair system, and whether it's the Model 1 and Model 2, or perhaps a combination of the two.
Well, thank you. We have time for one more question. Why should this machine learning approach be fruitful? It can only learn from the data that already exists. So where is the novelty?
Right. So the novelty is in the sense that the machine learning algorithms can only work on very large data sets that no human being can actually make sense of. Moreover, traditional statistical methods of linear regression or logistic regression can only look at linear correlations. For example, if A is correlated with B and B is correlated with C, we can say that A is correlated with C. So this is a case of linear correlation. But machine learning can find things that are perhaps non-linearly correlated. We simply don't understand what it is because the mathematics of it has not yet been developed. There may be non-linear relationships hiding in large amounts of data, and we do not have direct logical methods of analyzing them, which only some hidden properties of the machine learning algorithms can find. In other words, supervised machine learning is able to find relationships where individual parts combined together to make something that is more than the sum of the individual parts. And that's where the potential for such work lies. It is not that the data are novel, the data are not novel, but by analyzing the data, we get novel insights. And that is the novelty of this work.
Well, I would like to once again thank Dr. Ray for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through June 15th, 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.